On my last video, I discussed about the carbohydrates. Okay, what are the types of carbohydrates, functions of carbohydrates, okay, polysaccharides, monosaccharides and all. Now in today's class, we will going to discuss about the nucleic acid. Okay, nucleic acid is the second most important biomolecule, okay, that is required for most of the your class 11 and 12, as well as in your medical courses, as well as in your BSc and MSc courses. Okay, so nucleic acid is also one of the type of biomolecule that is present in a living organisms. Okay, and there are generally two types of nucleic acids are there. One is your DNA, okay, and another one is your RNA. These DNA and RNA, these are the two nucleic acids that specifically works for a life. Okay, that means for a life, for your from one organism to another, for your reproduction, to form a particular cell, to form a particular offspring, right, to form your sperms, ovas, okay, we require the DNA and RNA things. Okay, without the DNA and RNA things, this your reproduction and all and your life will not be possible. Okay, this DNA and RNA, it is present in all organisms, all living organisms. This DNA and RNA is present. Say for example, this DNA is present in your plants, okay, this DNA is present in animals, this DNA is present in your animals, this DNA is present in viruses also. Okay, the DNA and RNA is present in viruses also, in plants, animals, and that means in every living organisms this DNA and RNA is responsible. I mean it is present for their life to be happen. Okay, and RNA is basically discovered by Friedrich Csir. Okay, in some competitive exams these questions might come that who discovered this nucleic acid. Remember, the DNA and RNA nucleic acid is first discovered by Friedrich Csir. Okay, it was discovered by Friedrich Csir in a bandage, actually in an infected bandage. This DNA and RNA is discovered. Okay, I mean their structure is not discovered yet, but this nucleic acid word, okay, is basically coined by this scientist Friedrich Csir, who found this DNA and RNA in a bandage, okay, in a infected bandage or in a blood containing bandage, infected blood containing bandage. He discovered this nucleic acid, this DNA and RNA, and later on their structures are given by various types of scientists. Okay, and the most famous structure, that is the double helix model, okay, or the BDNA model is discovered by, actually this discovery of DNA is basically your DNA structure is basically done by your okay, so actually this DNA and RNA structure, okay, so since they are this nucleic acid was discovered by your Friedrich Csir, but their structure of DNA and RNA was discovered by other scientists. Okay, and the most famous conspiracious structure of your DNA and RNA was given by the pioneer of their DNA structure was basically Rosalind Franklin. Rosalind Franklin, she discovered this DNA structure, double helical structure, but later on Rosalind Franklin, let me tell you the name. Let me tell you the name. Rosalind Franklin, she was a lady, okay, and she discovered, she was a pioneer for the discovery of the double helical structure of DNA. But later on her theory was basically not I cannot say it is stolen, but somehow her data, I mean इनका डाटा किस एक और दो साइंटिस्ट था, फेमस साइंटिस्ट वाटसन एंड क्रिक, दे like take her data, okay, or they somehow manipulate her data and published their work, okay, and published their work, and also they got Nobel Prize for their DNA double helical structure, okay. And Rosalind Franklin, that lady, died, okay. And these two scientists were Watson and Crick. Watson and Crick. These are the two famous scientists, Watson and Crick, okay, who actually got the Nobel Prize for their DNA double helical structure, okay. But previously it was the pioneer was Rosalind Franklin. She died in some accident, I don't know about that, but she died. She did not got any Nobel Prize for that, and the Watson and Crick got the Nobel Prize for their double helical DNA structure, okay. Remember these two scientists name. It was first found by Rosalind Franklin, this DNA structure, not nucleic acid, remember, okay. Nucleic acid was discovered by Friedrich Mixture, not Watson and Crick, not your Rosalind Franklin. But later on their structure of DNA was basically given by your Watson and Crick and Rosalind Franklin, okay. So remember this name. And Watson and Crick, they got the Nobel Prize for their double helical structure of DNA, and they give the B-DNA structure. They give the B-DNA structure, okay. They give the B-DNA structure, okay. They give the B-DNA structure, okay. Now coming to their definition of nucleic acid, okay. These nucleic acids are basically composed of this nucleic acid is basically composed of polynucleotide. They are polynucleotide. They are generally polynucleotide in nature. Polynucleotide. They are polynucleotide in they are polynucleotide, okay. These nucleic acids are basically polynucleotides, okay, that is present in both DNA and RNA, okay. This nucleic acid means it is a polynucleotide, okay. That means presence of so many nucleotides one upon another in both the DNA and RNA, okay. Understand? They are polynucleotide. Now coming to their function of DNA RNA. अभी इस न्यूक्लिक एसिड का फंक्शंस क्या है, ठीक है. DNA RNA का फंक्शन क्या है, क्यों DNA RNA चाहिए, ठीक है. So the function is that the first function of the DNA and RNA is that the first function of DNA and RNA is that what are the function or what is the purpose what is the function of your DNA and RNA. The first function we can say, the first main function, this DNA and RNA, it has a hereditary role or hereditary material, okay. It has a hereditary material. That means This DNA and RNA passes information from parents to the offsprings. That means from father to the from father to the son, or to the mother to the son and all. Okay, that means transfer of genetic information from one generation to the next generation is basically done by your DNA and RNA. Okay, and what does it mean? Say for example, a monkey. Okay, when a monkey reproduces, it forms another monkey, right? So how the egg will know that it will be a monkey? Okay, the egg will know it will be monkey by their DNA codes. Okay, and again there is another example. Say for example, my hair color is black, so my son's or my daughter's hair color will also be a black in color. Okay, so that character, that traits, okay, is basically transferred by DNA and RNA in a particular organisms. As well as in case of your plants, okay, the plant's character, the plant's fruit, okay, all the informations will be passed from one plant to their offsprings will be also done by your DNA and RNA. Okay, past of antibiotic resistance in a particular bacteria. ठीक है, बैक्टीरिया के पास क्या होता है? एंटीबायोटिक रेजिस्टेंस होता है in some bacteria in their plasmids. Okay, so that antibiotic resistance will also transfer by DNA and RNA. So this is one function of DNA, is that the DNA is a hereditary material. Understand? Fine. Next important function of DNA is that it codes for a protein. That means protein formation करने में भी DNA responsible है. Protein formation, DNA and RNA is responsible. Protein formation, most important function of DNA and RNA. Okay, to form a particular protein in our body, we require the DNA. To form, say for example, to form collagen protein in our body, to form a particular enzyme, say for example salivary amylase, okay, we require DNA. Okay, the DNA has a specific codes that tells our body to form collagen and to form your salivary amylase enzyme. Okay, so all the informations are basically stored in the DNA. Okay, so basically DNA, RNA are the softwares, okay, or is a CPU of the human body that tells your body to perform this function, to perform these functions, to perform a normal metabolic rate in the body. All of the things were basically done by your DNA. Okay, if somehow there is a error in the DNA code, if somehow due to some external factors, if there is a error in the DNA code, it will lead to mutation. That means it will form a faulted protein, it will form a abnormal protein, or it will hamper your body's functions, normal function, due to their error in the coding process. Okay, so that is called as a mutation. Okay, so another function of DNA is that DNA क्या करता है? DNA प्रोटीन फॉर्म करता है बॉडी में. हमारा बॉडी में जितना भी प्रोटीन फॉर्म हो रहा है, जैसे कि हार्मोन्स हो गया, जैसे कि तुम्हारा एंजाइम्स हो गया, कोलाजेंस हो गया, ठीक है, सेवरल टाइप्स ऑफ पेप्टाइड्स हो गया, ऑल सेवरल टाइप्स ऑफ तुम्हारा receptors हो गया. So all of this will basically were formed by your DNA. This is one function, is that DNA will help in the formation of protein. Next important function of DNA and RNA is that it will help for formation of ribosomes also. Ribosomes all of you know. These ribosomes help in protein formation or protein translation. So this ribosome is basically formed by your rRNA. Okay, there are various types of RNA. I will come to the later. Okay, it will form by your RNA molecule, rRNA molecule. That means for the formation of ribosomes, we also require RNA molecule. Okay, RNA molecule or your DNA molecule. Okay, fine. Next function, this DNA is your development of immunity, development of immune functions, development of a particular cells, okay, and also helps, this DNA helps in apoptosis. Apoptosis means cell death, okay, मतलब एक cell जब, every cell has a particular life. Say for example RBCs has 120 days, okay, so after 120 days the RBC must die, okay, so that dying process is called as the apoptosis, and it is coded by your DNA, or it is coded by a particular protein that is also coded by your DNA, okay, that is called P52. There is a protein which will code for apoptosis, okay, so then the cell will die at a particular time. Every cell in our body has a specific time for their growth and for their death, okay. If the cell is not able to die, okay, अगर वो सेल मर नहीं गया तो क्या होगा, there will be abnormal mass of cell, एक के ऊपर फॉर्म होता रहेगा, उससे क्या होगा, ट्यूमर फॉर्म होगा, और वो ट्यूमर के वजह से कैंसर हो सकता है, ठीक है. इट मे कॉजेस कैंसर ऑल्सो इफ द DNA इज ए डैमेज, और इफ देयर इज ए प्रॉब्लम इन द DNA, okay, it will lead to cancers. Understand? So this is all about general function of DNA and RNA. The first function, the most important function is the hereditary unit of life, okay. It is a functional and hereditary unit of life, okay. It helps in protein formation or protein coding, okay. It helps in formation of ribosomes. It helps in the for your body to develop immunity, to fight infection, to develop specific organs and all, okay. Then it will all the basic difference, okay. I will I'm telling you two difference between some not only two, sorry, it will also cut in two difference between, two or more difference between DNA and RNA, ठीक है, कि इन दोनों के बीच में difference क्या है, to what are the difference between DNA and RNA, okay. अभी DNA and RNA में difference क्या है, okay. So there are some basic differences in DNA and RNA. One of the main important of DNA and RNA is that it is double stranded, okay, double stranded. Most of the DNAs are double stranded in nature, okay, and it is single stranded, okay. This is one difference of DNA and RNA. Second difference is that DNA contains deoxyribose sugar, deoxyribose sugar, okay, and your RNA contains ribose sugar. DNA contains deoxyribose sugar and RNA contains ribose sugar. Next one is your DNA has nitrogenous bases, right? So DNA has adenine, thymine, guanine and cytosine. I will discuss this, what is adenine, thymine and guanine, in my coming videos, okay. And your RNA contains adenine, uracil, guanine and cytosine, okay. This T is absent, that means thymine is absent in case of RNA, okay, and uracil is absent in case of DNA. This is another example of DNA, difference of DNA and RNA. That is, thymine is absent in case of RNA, and uracil is absent in case of DNA, okay. Why this is happening, I will discuss this, okay. Next, coming to the next difference is that the DNA obeys, it will obey Chargaff rule. Most of the double stranded DNAs obeys Chargaff's rule. It is a rule. I will discuss this, what is Chargaff rule, in my coming in next point. And this RNA does not obey Chargaff's rule, okay. It does not obey Chargaff's rule. This RNA does not obey Chargaff's rule, but DNA obeys Chargaff's rule, okay. This is another difference between DNA and RNA, okay. Another example of DNA and RNA is that DNA is present, most of the DNA is present in nucleus, okay. Nucleus, it is present. all the DNAs are present in your nucleus, okay, and RNAs are not present in nucleus, okay. These RNAs are not present in nucleus. Most of the DNAs are present in nucleus and RNAs are not present in nucleus. So here are the 5 different. Now coming to their structure, that means how a DNA RNA is made up of, okay. Okay. 5 dash, okay. Five dash, okay. So this is the typical structure of okay. So see, this is your structure of DNA, double helical structure of DNA, okay, and it is a nucleic acid, okay, and this nucleic acid, it is a polynucleotide, right? It is a polynucleotide. This this DNA is a polynucleotide, okay. That means it is made up of so many nucleotides, okay. That means there are so many nucleotides is attached in DNA in one upon another, okay, via a bond called phosphodiester bond, okay. And one nucleotide is basically made up of 3 components. One is a sugar molecule, a pentose sugar, okay, a pentose sugar, okay, then nitrogenous base, okay, nitrogenous base and a phosphate group, nitrogenous base and phosphate group, okay. These three things will form a nucleotide, a nitrogenous base, a pentose sugar, okay, and a phosphate group, okay. And for a pentose sugar, it can be either deoxyribose sugar which is present in DNA, okay, or it can be your ribose sugar which is present in your RNA, okay. The pentose sugar can be of 2 types in a nucleotide. One is your ribose sugar which is present in RNA, and one is your deoxyribose sugar that is present in your DNA, okay. Then we have the nitrogenous bases, okay. And what are those nitrogenous bases? The nitrogenous bases are adenine, okay, adenine, then thymine, okay, thymine, then cytosine, okay, then uracil, okay, adenine, thymine, cytosine, guanine, and uracil, okay, and uracil. These are the nitrogenous bases that is present in your both the DNA and RNA, okay. The exception is that this U, that means the uracil, is present in RNA, okay, and thymine is present in your DNA, not in RNA. Understand? These are the nitrogenous bases that is present in a nucleotide, okay, adenine, thymine, cytosine, guanine, uracil, and adenine, okay. And we have a phosphate group, okay. What is a phosphate group? What is the role of a phosphate group? The phosphate group will help in the attachment of one nucleotide to the next in a polynucleotide, that means in a nucleic acid. The phosphate group will help in the attachment of one to another. That is the role of phosphate group, okay. So this is about your nucleotides. That means a nucleotide is made up of basically three things: a pentose sugar, a nitrogenous base, okay, and a phosphate group. These three things, pentose sugar, nitrogenous base, and phosphate group, will combine to form a nucleotide, and so many nucleotides will combine to form a polynucleotide, that is a nucleic acid, okay. All of you understand? If you guys have any doubt, the comment section is free for all, okay. You guys can ask me there. These three things are joined with each other, this phosphate group, this nitrogenous base, and sugar. Remember, the nitrogenous base, okay, is always bound with a this nitrogenous base is always bound with a sugar molecule. The nitrogenous base is always bound with a sugar molecule, and this sugar molecule, it is attached by a phosphate group. These three will combine and form a nucleotide, okay. These are the basic concept, okay. Without knowing the basic concept, you cannot go to the actual structure of DNA and RNA, okay. So this nucleotide is basically formed of three things: nitrogenous base, a sugar molecule, and a phosphate group. This nitrogenous base is attached with a sugar molecule, and the sugar molecule is attached with a phosphate group, okay. And when another your nucleotide when joins, it will join via this phosphate group, okay. That means there will be another nitrogenous base will be there. There will be another nitrogenous base. That means there will be another your nucleotide. It is attached with a sugar molecule, okay. It is attached with a sugar molecule, and this sugar molecule is attached with the phosphate group, okay. This sugar molecules is attached with a phosphate group. In this way, the nucleotides will join upon one another, okay, in a nucleotides, okay. They are joined in this fashion, remember, okay. Okay, in this way the nucleotides are attached with one another by the help of this phosphate group, okay. And remember, the nitrogenous base is always attached with a sugar molecule. The nitrogenous base always attached with a sugar molecule, okay. And the combination of this nitrogenous base plus sugar is called as nucleotide, nucleoside is called as nucleoside. The combination of nitrogenous base in sugar is called as nucleoside. Okay, and when there is the attachment of phosphate group with a nucleoside, it is called as nucleotide. Okay, so in my next class I will discuss about the nitrogenous bases, the sugar molecule structure and the phosphate group. Okay, so till then you guys see my video for the introduction of DNA, RNA, their function, and about the nucleotide. Okay, so